Hello my dear students, welcome back to my channel. Now in this session I am going to discuss about two small topics. One is artificial hybridization, another one double fertilization and triple fusion. So let us now start with what is artificial hybridization. This is the most important method for crop improvement where a desirable pollen is taken and is transferred to the stigma of a female parent. And this helps in production of hybrid varieties with all desirable characters. So there are different steps. So let us try to list down what are all the different steps. First one is selection of parents. Second is called emasculation. The third one is bagging. Fourth, artificial cross-pollination, and then rebagging. Selection of recombinants or desirable progeny. Now if we go to the first one, selection of parents, the most important thing here is germplasm collection. Now what is germplasm? Germplasm is presence of all diverse different alleles of a particular plant species. So this germplasm collection is done in various institutes. Like for example, we have IRRI, International Rice Research Institute, located at Philippines, that has a germplasm collection of about 10,000 rice varieties. So in this way, every institute has germplasm collection of different plants. So we can bring the germplasm from that particular collection, and then we can use them as the parents. So once the parents are selected and then two parents are available, that is male parent and female parent. I already gave you the definition of what is male and female parent. Female parent is which receives pollen grain, desirable pollen grain, even if it can be a bisexual also, but it receives a pollen grain. That is called as a female parent. So first step here, next we can take after the plants are collected, selection of parents. What is next one is called emasculation. In emasculation is removal of anthers from a bisexual female parent. Why it is done? In order to prevent self-pollination. In order to prevent self-pollination. And emasculation, it is not required for unisexual parents. If the female parent is unisexual, then emasculation is not required. Let us now go to the third step, that is called. Now let us start with what is bagging. After emasculation, the female flowers are covered with a polythene bag. Female flowers, completely the inflorescence will be covered with a polythene bag. So what is the use of this? Bagging will prevent any unwanted, it prevents any unwanted cross-pollination. It prevents unwanted cross-pollination. That is the purpose of bagging. Now after bagging, artificial cross-pollination is done. So carefully the pollen grains are collected from the male parent and it is dusted on the stigma with the help of a brush. So here requirements are forceps and brush. So these two are requirements. So what are requirements here? For artificial cross-pollination, forceps and brush are required. So they are removed with the help of forceps, the anthers are removed and pollen grains are taken, and with the help of, they are transferred to the brush, and with the help of brush they can be easily dusted on the stigma. That is called as artificial cross-pollination or hybridization. After that again rebagging is done. So what is the purpose of rebagging? So rebagging is done in order to ensure fertilization. Rebagging is done in order to ensure fertilization. That means there is proper fertilization is taking place. That is the purpose of rebagging. Then last one, selection of recombinants or desirable progeny, selection of recombinants or with desirable progeny. So after the fertilization process takes place, the progeny called as F1 progeny. F1 progeny is selected. F1 progeny is selected for superior characters. It is selected for superior characters. And then this F1 progeny will be subjected to self-pollination because the characters are completely in heterozygous condition. So this F1 progeny directly cannot be used for cultivation. So what is next step done for at least for 3 to 4 generations? This is subjected to self-pollination. And then the importance of self-pollination is homozygosity is reached. Due to self-pollination, there will be homozygosity will be developed. Homozygosity is the results in stability of characters. If characters are in heterozygous condition, they are less stable. If they come under, if they become homozygous, then it is more stable. So only after cultivating it for 3 or 4 generations, then they are given to any further cultivation. So these are the different steps in artificial hybridization. So this will be a very small topic which is in the NCERT book. The most common type of questions which you can get is importance of each step, that is one you can get, and sequence of these steps. So the same sequence you have to follow. That's why first I listed the sequence. So you are going to get about the sequence of the steps. Okay students and other subscribers, let us now end the topic here. Let us now conclude my video. I want to conclude here. In my next session, next video, I am going to discuss about double fertilization and triple fusion. And also, there are some questions related to that. Both together, I want to discuss in the next topic. So, I hope you have followed the video from beginning to the end. Thank you very much. Please do subscribe.